Our next topic is called gene interaction. Gene interaction is remember I was talking about how genes act in like recipes? So this is really not completely different from that idea. Two genes produce gene products, and those two gene products interact in a recipe, like the butter and the sugar and the flour and the eggs. So gene interaction really extends to all genes interact at some level, but we're going to handle it. We're going to talk about how do two separate genes interact. So attacking this concept at the very basic level, just to start. So in these examples, you'll just see two genes interacting, and we can expand that in a course that you may take, more advanced course, but just the basic idea here. So what gene interaction is, and like we said, this is something Mendel didn't figure out, but it turns out that some genes produce gene products that have to interact with each other. And so, for example, you have these peppers, and there's a gene for the red color. They're calling that gene Y. And then there's a gene that says whether or not there will be color, or a lot of color. So big C would be a lot of color, little c would be not very much color, for example, less color. So the C is like the intensity of the color, and the Y is the red. And so what you can have here is, let's say you cross a red pepper with a cream pepper. And if you follow the genetics of it here, you can see why you would get this genotype. And the pepper looks completely red because the big Y is the dominant red allele and the big C is the dominant for lots of color. But then when you self-fertilize this guy, you get not just red and white anymore, you get some in-between colors. And what this is the interaction of these two genes in different genotype combinations. So you do get a 9 to 3 to 3 to 1 ratio. You get 9 that are red, and you get 1 that's cream. And then in the middle here, you get some that are peach and some that are orange. And the peach ones have the red allele, but they have no color on the other allele. So it's like they have an allele that produces a bright red, and then on the other hand, they have a genotype that should say not very much of it, not very much color. On the orange one, it's kind of the flip side. They have no red allele, but they have an allele for a lot of color, so then that comes out in a different way. So it's the interaction of two genes, not two alleles with each other, but two entire genotypes interacting with each other. So gene interaction is a little bit like dominance and recessiveness, but instead of dominance and recessiveness as an interaction between two alleles of a single gene, instead it's an interaction between two genotypes of two different genes. I know, it makes your head want to explode, right? An extension of this is called epistasis. Epistasis is a type of gene interaction where a genotype at one locus completely prevents expression of a genotype at the second locus. Remember, the locus is the location. So one gene, and not just the gene itself, but the particular genotype of that gene has the ability to completely block expression of another whole genotype. The most common example of epistasis is Labrador retrievers. They have different coat colors, and to put it very simplistically, if you think about the black allele as being dominant to the brown, those alleles are alleles of the same gene, black and brown. But then there's a whole nother gene that says whether or not there will be color at all, kind of like the peppers. There's a C, and if you have a big C, that means there will be color, and if you have a little c, that means there won't be much color. So the yellow ones, what we call yellow labs, have to have a little c, And that blocks, even though they can have a baby that's black themselves, they might have the black allele, but they have the little c, which says no color, or maybe not very much color. And so little c blocks expression of the black or the brown allele, which is a different allele. So for example, let me see if I can get a pen here. Okay, so very much like the example with the peppers, remember the red and the cream peppers, the Labrador Retrievers would have two different genes, at least two different genes, although now we think there's probably more than two. And maybe you could think of the locus B representing the color, black or brown. Excuse me. Where black is dominant and brown is the recessive, so to speak. So big B is black, little b is brown. But then you have a whole nother gene where big C means there will be a lot of color, and little c means not much, if any, color. Little c, no color, or not very much. I'm just going to put no color. Sorry, handwriting with the mouse is not good. Now, so what you end up with is that a yellow lab could very easily be homozygous for the black allele. I mean, it's possible. But then homozygous for the no color allele. And that would end up with a yellow. But then there's different shades of yellow, so we actually think there's probably a whole nother gene. Maybe you could call it gene D or E or F. I mean, so that might indicate different shades of these colors. But at least you get a concept that if you're little c, that's going to mean no color, even if you're big B also. The little c will rule or sort of dominate over the other gene. So that's epistasis, where the little c blocks expression of the B locus. Okay, moving on to a different topic, cytoplasmic or extranuclear inheritance. You may remember from the endosymbiont theory or the endosymbiont hypothesis that mitochondria and chloroplasts have their own chromosomes. They have their own ribosomes. And so there are genes on those chromosomes, and you can have mutations or alleles of those genes, and there can be mutant phenotypes. Any mutant allele must be inherited from the mother because all of the cytoplasm in the zygote is maternal, and cytoplasm includes the mitochondria. So the mother's mitochondrial genes will be passed to the child. Virtually none of the father's mitochondrial genes will be passed to the child, and this picture here shows you why. You have a vast difference in the contribution of the zygote's cytoplasm. There's a big difference between how much the egg contributes and how much the sperm contributes here. There aren't very many disorders that are associated with alleles on the mitochondrial chromosome because, as you can imagine, a defective mitochondrial chromosome is going to be very lethal to a cell. Defective cellular respiration is ultimately what we're talking about. But there is a disorder called MRF. It's pretty rare, but it's myoclonic epilepsy with ragged red fibers, and it has to do with a defective mitochondrial gene. Mitochondria are not segregated perfectly in cell division. They're randomly segregated, so in order for a cell to end up with a high proportion of defective mitochondria, you would have to have a series of cell divisions that ultimately, just by chance, yields a cell that has either all or predominantly all mitochondria that are defective. That's the only time you would really probably see the phenotype. All right, changing topics again, imprinting. Imprinting is a specific case of what we call epigenetics. Epigenetics means chemical modifications directly made to a chromosome, typically attachments of certain functional groups or molecules to the chromosome that influences the expression of the genes on that chromosome. So imprinting is a special case of that. It's when maternal or paternal gene regions are turned off during the formation of gametes. Because these gene regions are important for the early stages of embryogenesis, what you end up with is an egg that cannot develop into an embryo and a sperm that cannot develop into an embryo, but rather when both genomes fuse together, you have a situation in which only in that case the embryo has all the genes it needs to develop. Imprinting isn't permanent. It is removed. It is a chemical molecule, usually a methyl group that's attached to a chromosome, so it can be removed from a chromosome when that chromosome it goes through meiosis. So here's an example of imprinting in a gene that's known to be imprinted. It's a gene called IGF2, and of course everybody has 2 copies of chromosome 11 in every cell. The chromosome you got from your mother was imprinted, meaning it has methyl groups attached to it so that it cannot be expressed. The IGF2 you got from your father is active. So in the end, every cell in your body actually only expresses your father's IGF2. And so IGF2 is insulin growth factor, and it has to do with growth, size, weight control, those types of things. And it's going to be more influenced. That particular gene is going to be coming just from your father. Some proteins are ultrasensitive to slight temperature variations. Anytime you have an allele that creates a protein that's ultrasensitive to temperature variations, it's called a temperature-sensitive allele. And an easy example of this are the enzymes which produce the pigmentation in these kinds of rabbits or Siamese cats. These are temperature-sensitive alleles. They can only function at the cooler temperatures like the nose, the ears, and the hands and feet, arms and legs. But the body temperature, the core body temperature, is too high and the enzymes cannot function. So you get no pigmentation in the main body, a main part of the body, but you get pigmentation because the enzymes can function in other parts. And we see the fruit fly presented in this chapter. You would need to focus mainly on contributions to genetics when we look at these model organisms. I don't expect you to know all these details, but I do expect you to remember the fruit fly was important for sex linkage, for example, because you remember Thomas Hunt Morgan. And so for now, I would expect you to know that. But it's been a very important model organism for a lot of things. But for right now, if you can connect the fruit fly with Thomas Hunt Morgan, that's a good place to start. The genome of the fruit fly, you don't need to memorize this, but it's very small. There's only 8 chromosomes per cell. But the number of genes is probably about 15,000. This says 14,000. And something around there. And so, humans only have about 22,000 genes. So a lot of the genes that fruit flies have will be relevant, have a homology to something in the human genome. So that's why in a lot of ways, they've been helpful to study them. So this chapter we've covered many of the extensions and modifications of Mendelian laws, but we haven't covered everything. We'll see some other things a little later in the course, and there are some things we won't talk about at all, but we've had a good solid basis here for some of the more common exceptions to Mendelian law. But genetics isn't just so straightforward. This is chapter 4 really explains why Punnett squares really just don't solve everything or don't predict everything, and therefore genetics is not as simple as you may have wished. And so I said sorry. Make sure when you're studying chapter 4 you focus on each of the terms that describe the different phenomena, the definition of the phenomena, and then the example that I gave. All right, that's how I would study for this, because there's a lot of things in this chapter, and I think it's the most difficult chapter in the whole course, in a lot of ways, because there's so much information here. All right, so please let me know if you need any help or have any questions.